Imagine peering into the void and seeing shapes that aren't really there. Our brains are the ultimate illusionists, conjuring reality from thin air. But what if the secret to these mind-bending tricks lies in a special group of brain cells? Dive in as we explore groundbreaking research that could redefine how we understand perception. In a fascinating leap forward, scientists led by Shin have uncovered a type of neuron that plays a starring role in our ability to perceive illusory contours, those optical illusions where our eyes detect edges or shapes that don't actually exist in the visual world. Think of the classic Kinesa triangle. You see a white triangle popping out from three Pac-Man-like shapes, even though no triangle is drawn. Earlier studies had teased the existence of such cells, but Shin's team has now demonstrated that these aren't just quirky outliers. Instead, they're a well-organized, vital subgroup within the primary visual cortex, the brain's first stop for processing what we see. For beginners, the primary visual cortex is like the brain's main hub for raw visual input, and these neurons help fill in the blanks, completing patterns that make sense of incomplete information. But here's where it gets controversial. Are these neurons the puppet masters behind our perception, or just bit players in a larger show? Shin explains that these cells, dubbed IC-encoders for illusory contour encoders, actively drive what's called local pattern completion right in the primary visual cortex. This process allows the brain to infer and construct missing parts of an image, which researchers speculate ties directly into how we experience illusory contours. Adesnik, another key researcher, adds that their experiments showed these neurons are causally linked to this completion, meaning they don't just respond to illusions. They help create the neural patterns that mimic real perception. Now, while this sounds like a breakthrough in understanding how we see the unseen, there's a catch. The study didn't prove that mice actually perceived these illusions when the neurons were artificially activated. Adesnik clarifies that their focus was purely on the neural side. No behavioral tests were conducted to check if the mice behaved as if they saw the contours. All they observed was that stimulating these neurons produced brain activity patterns that matched those seen during natural illusory contour perception. It's like watching the orchestra play the notes without hearing the full symphony. Shin acknowledges this limitation, noting that the optogenetics technique they used, a method to control neurons with light, could only target a small number due to technical constraints. IC-encoders are relatively scarce and spread out across the brain's layers, so activating just a handful might not be enough to trigger full perception. And this is the part most people miss. Could expanding the stimulation to, say, 3 to 10 times more neurons finally make the mice see illusions without any visual cues on a screen? That's the exciting frontier ahead, as Adesnik outlines. Future experiments could involve photostimulating larger groups of these neurons to elicit behavioral responses, such as mice reacting to non-existent shapes. Right now, optogenetics is limited, but imagine if we could recruit hundreds or thousands of these cells. Might we unlock the ability to generate perceptions from nothing? The team is eager to pursue this, potentially bridging the gap between brain activity and actual experience. This research, published in Nature Neuroscience in 2025, DOI 10-1038 S41593-0255-5, opens doors to deeper questions about consciousness and reality. But is it ethical to manipulate perception this way, or does it blur the line between what's real and what's artificially induced? What do you think? Could these findings one day help treat visual disorders, or do they raise concerns about creating false realities? Share your thoughts in the comments. Do you agree that these neurons are key to illusions, or is there a counterpoint I'm missing? Let's discuss.